Assalamualaikum warahmatullahi wabarakatuh. Salam Om Swastiastu, Namo Buddhaya, salam kebajikan dan salam sejahtera untuk kita semua. Selamat pagi dan selamat datang di acara webinar ImmunoFluorescence Technical Update for Research and Diagnostic Application. Acara webinar ini merupakan kerjasama antara PT Vantagebio Scientific Solution dengan Vector Laboratories. Kami ucapkan terima kasih kepada Vector Laboratories sehingga presentasi ini dapat terselenggara. Saya Fitri Rahmasari dari PT Vantagebio Scientific Solution. Dalam presentasi ini akan dibahas mengenai hal-hal yang perlu diperhatikan serta technical problem yang sering dijumpai dalam aplikasi ImmunoFluorescence. Sebelum presentasi dimulai, kepada para pemirsa, untuk dapat memberikan dukungan pada channel kami, dimohon untuk mensubscribe dan bunyikan lonceng untuk channel kami. Dukungan Anda sangat berarti dalam memotivasi kami untuk membuat video maupun presentasi selanjutnya. Presentasi kali ini akan dibawakan oleh Byron dari Vector Laboratories. Byron merupakan Senior Technical Service Specialist di Vector Laboratories. Byron memperoleh gelar Master of Science dari Biomedical Engineering di University of Miami, Florida. Sementara untuk Bachelor Science diperoleh dari Bioengineering di University of California, Berkeley. Mari sama-sama kita simak presentasi dari Vector Laboratories.
Other technologies such as immunohistochemistry. For one reason, IF is easier to visualize for multiplex staining. When you have two antigens that are co-localized or in the same cell compartment, it's easier to mix the different colors. Let's go over the basics of this webinar. First, we'll go over the workflow optimization for immunofluorescence. Next, we'll discuss autofluorescence background. And then we'll go over detection techniques, mostly one-step and two-step methods. Last, we will discuss the preservation of the signal intensity from the fluorophore. So for the scope of this webinar, we will focus on two types of microscopy, epifluorescence and fluorescence confocal microscopy. The two primary formats will be fixed cells on a monolayer on a cover slip. The second one is a thin cut tissue section, probably 5 microns thick. So the purpose of immunofluorescence is to use color to identify and differentiate different target antigens and cellular components. This means using antibodies to bind to antigens in the cell or tissue section and visualizing the site of localization. You can see in this image on the right, a double label. The red color represents cytokeratin, a antigen. The green color represents desmin, another antigen. And the blue is a DAPI counterstain that stains the nucleus for visualizing the architecture of the cell. Next, I will discuss the basic concept of immunofluorescence. Essentially, there are fluorescent molecules that absorb one wavelength of light and emit the light at a higher wavelength. The reason for this is that the emission light falls to a lower energy state. And based on this, there are different emission and excitation spectra for each fluorophore. You can see on the right in the graph, the image of excitation and emission spectra. And this is different for each fluorophore. And this difference is the reason why you see different colors from the visible to far red range. This could be green, red, or far red colors, for example. And the difference between the excitation and emission wavelength is called the Stokes shift. and this value is different for each fluorophore. Next, we will discuss optimization steps for immunofluorescence. The first step is tissue preparation. These initial steps have a big influence on the final signal intensity from the fluorophore. For fixation conditions, there are a couple considerations. You can use a formalin-fixed paraffin-embedded tissue section or a acetone-fixed frozen section. Each one has different advantages and disadvantages. For the formalin-fixed tissue sections, they are stored at room temperature before staining. For the frozen sections, as implied, they are stored at -70 degrees Celsius. The next consideration is antigen retrieval. This will affect the signal intensity downstream. Antigen retrieval is typically required just for formalin-fixed tissue sections and not acetone-fixed frozen sections. And the optimal antigen retrieval based on pH, incubation time, and temperature vary from antigen to antibody combination. You would have to empirically test which antigen retrieval method is optimal. Here's an image of prefixed frozen material that has undergone antigen retrieval. If the frozen section has been formalin fixed, you may still perform antigen retrieval, but at a lower temperature and less time. Next, we will discuss the next couple steps in the immunofluorescence staining protocol. Step 2 is the protein block. This helps to minimize background from Fc receptors and charged molecules. You can use normal serum, BSA, and other blocking agents. Step 3 is the application of the primary antibody. Just make sure you titer the primary antibody for each application. Step 4 is the secondary detection reagent, which could be various one-step and two-step methods, such as a fluorophore conjugated secondary antibody. Step 5, if necessary, you can quench autofluorescence from your sample and really increase the signal-to-noise ratio. The last step is mounting before cover slipping. This is important because all fluorophores eventually fade over time due to oxidation. So it is important to have an anti-fade mounting medium to preserve the fluorophore for archiving. I'll briefly go over positive and negative controls. Positive controls could be the known tissue sample, and it should be prepared in the same way as the test sample. For negative controls, there are various kinds. You can use a no primary control or a autofluorescence negative control. This means you just omit all the detection reagents and see how much background you can see. And the purpose of all these steps is to increase the sensitivity and retention of the fluorescent signal so you can have the most maximal signal intensity. So I've just mentioned positive and negative controls. So based on your negative control, you may see autofluorescence background. And this is due to noise you see in the image below. So I'll define autofluorescence background. It is fluorescence endogenous to the tissue. This could come from various sources, such as lipofuscin, which is an aging pigment. Other sources could be aldehyde fixation, such as formalin, or endogenous tissue elements, including red blood cells and collagen elastin proteins. And the problem with background fluorescence is that it lowers the signal-to-noise ratio. There are various methods to decrease the amount of noise so you can see the signal clearly. Okay, so the next, how do I reduce autofluorescence background? The first way is to optimize microscope settings. If you're using a confocal microscope, you can illuminate the slide with LED light and quench the background fluorescence. Make sure you do this before you apply any fluorophores in the detection steps. Next, select optimal fluorophores. Typically, autofluorescence is most common in the green channel, and some people can choose a far red fluorophore to minimize this autofluorescence. However, even in the far red channel, there is autofluorescence present between 600 and 700 nanometers. Third, you can select appropriate quenching reagents. Based on the result of your negative control, you can determine whether the autofluorescence is from lipofuscin, the aging pigment, or from non-lipofuscin sources. That could include the aldehyde fixative or red blood cells. And that will determine which quenching reagent you should use. And you can see below just various emission and excitation spectra of fluorophores across the visible spectrum. Next, I will briefly go over a previous investigation on reducing autofluorescence. In this image, you can see the untreated human tracheal tissue section with some green background autofluorescence. There are 3 different masking treatments here to minimize the background. First is sodium borohydride that targets the aldehyde fixative. Sudan black B is commonly used to quench lipofuscin sources, and Aerochrome black T. These methods use a masking method to absorb incident radiation. And just to clarify, these methods do have pros and cons for certain tissues. So next, we'll go over the various immunofluorescence detection steps. The first method is a one-step detection. You first apply your primary antibody onto the cell monolayer or tissue section. This will bind to the target antigen in the sample. Next, you apply a fluorescent secondary antibody that binds to the primary antibody. This allows you to visualize the target antigen at the site of localization. However, this is a one-step method and not very sensitive compared to two-step. Next, I will go over a few two-step methods that really increase the sensitivity of your detection. This is more useful for target antigens that are weakly expressed, or if you don't know the expression level. So first you apply the unconjugated primary antibody, and then the biotinylated secondary antibody, which is biotin conjugated, will bind to the primary antibody. Next, you will apply a fluorophore conjugated strep avidin or avidin. The fluorophore will be visualized in the target sites because of the strong non-covalent bond between strep avidin and biotin. The avidin has 4 biotin binding sites that really bind strongly to biotin, and this is almost irreversible. So based on the previous slide, we'll build on that two-step detection system and really find another way to amplify the signal and increase sensitivity. As before we stated, the first step is the primary antibody, followed by the biotin conjugated secondary antibody, which is anti-mouse IgG here. The third step is a strep avidin fluorophore, such as fluorescein. And then the subsequent steps will help you to really increase the signal intensity by applying layers of additional fluorophore at the site of localization. So you'll first apply a biotinylated anti-streptavidin that binds to the streptavidin fluorescein. And then you will reapply the streptavidin fluorescein again, and that will increase the amount of fluorophore in the sample. This is an alternative two-step detection method, and this is non-biotin based. The reason for avoiding a biotin-based detection system is that certain tissues contain high levels of endogenous biotin. This could be kidney, spleen, or liver. And in these tissue types, you may want to consider this type of method. First, you apply an unconjugated primary antibody, such as a mouse IgG, and then apply an unconjugated secondary antibody, a goat anti-mouse IgG here, that binds to the primary. And then for the third step, you add a fluorophore conjugated tertiary antibody. In this case, it's a horse anti-goat IgG conjugated to dialite 488, which is a green fluorophore. So in this method, you're able to avoid the introduction of biotin and still amplify the signal with high sensitivity. This is useful for weakly expressed antigens. The next method is called pteramide signal amplification, or TSA. In general, this system can be used to really increase the intensity of the signal, probably more than the previous two-step method. So in this diagram, you'll see that the secondary antibody is conjugated to an enzyme called horseradish peroxidase, typically used for chromogenic detection. Then you can apply a pteramide. The pteramide is first inactive. It could be conjugated to Alexafluor 488 or biotin. And then once it is exposed to the catalyst, hydrogen peroxide, this enzymatically converts the pteramide to an active pteramide that allows the fluorophore to be at the site of localization. So this is a very useful technique to really boost the sensitivity of your signal for weakly expressed antigens or unknown expression. Next, I'll discuss fluorophores and the selection of various fluorophores. This covers the visible and far-red spectrum. On the left, you can see AMCA and DAPI in the blue channel. Some green fluorophores are dialite 488, FITC, and GFP. Then you'll go to the orange fluorophores such as Alexafluor 555, and then the red fluorophores such as dialite 594 and Texas Red. In the far red channel, you can use Alexafluor 647 or Sci5. Okay, and this is a fairly busy slide, but I'll break it down for you here. So on the right is a list of microscope light sources. Could be various laser sources such as argon ion, diode, or krypton argon. In this example, we'll focus on just krypton argon lasers. So this laser basically it shines a light at 488 nanometers and 568 nanometers. And this corresponds to the two fluorophore excitation spectra in the diagram, the fluorescein or FITC, which is green, and the Alexafluor 546, which is orange, red. And since the laser is near the peak of both the FITC and the Alexafluor 546, the excitation and the emission will be very strong intensity. And so this is very useful for double labels, a sequential double stain with two different fluorophores. You can see in the diagram, there is spectral overlap between the two two spectra, part of that Lexifluor 546 spectra overlaps with the green, and this affects the emission of the fluorophores and possibly result in some spectral bleed-through from overlap between the two spectra. So it is important to choose the correct filter cubes for your fluorophores. Make sure they really demarcate or differentiate the two emission spectra that you're using in your multiple label. Okay, let's summarize how to select fluorophores. So the first one is to select an appropriately colored fluorophore for your application. Just make sure that if you're using a multiplex IF system, that the fluorophores don't overlap too much. So a typical combination will be green and red, not orange-red. Next, make sure you match the fluorophore with the microscope setup. Based on the previous slide, the light source will determine which filter cubes you should select. For krypton argon, you might choose various excitation and emission filter cubes for your FITC and Lexifluor 546. The next point is about how different fluorophores generate different levels of intensity or sensitivity. For example, the FITC or fluorescein in the previous slide is a first generation fluorophore. And there are newer fluorophores with better photostability and less photobleaching. There could be Lexifluor 488 or Dialite 488. Just make sure you choose the fluorophore that suits your needs. And just to conclude, different fluorophores photobleach at different rates. So the FITC tends to photobleach faster than the Dialite 488. If there is endogenous green fluorescent protein in your sample, make sure you avoid any green fluorophores such as FITC, because they're going to cause background and interfere. Okay, next we will discuss how to preserve your signal. I mentioned previously that all fluorophores eventually fade over time. So it is important to use an anti-fade mounting medium. The anti-fade reduces the amount of oxygen that causes the quenching effect. So there are various homebrews that can be used as anti-fades. This includes N-propyl gallate and Movil Dapco. The recipes for these homebrews are available from various research labs. However, you can also use commercial sources, such as our own Vector Shield anti-fade mounting media. The Vector Shields come in a variety of flavors. It could be non-setting or non-curing, setting or curing, which tends to harden over time. You can also choose different vector shields with and without counterstains. The counterstains could be DAPI for nuclear counterstaining or phalloidin for actin imaging. And you can see in the image here, we focus on manual cover slipping. So you apply about 25 microliters or so of the Vector Shield anti-fade mounting medium onto the glass slide, and then cover slip over the sample. And then you can archive the slide for later imaging. So why do you need to use anti-fade media? Well, you want to reduce photobleaching of the fluorophore that occurs during specimen analysis. These images here, you can see the initial appearance is a green fluorophore with DAPI counterstain. So under 40x objective, you can see that with the anti-fade mounting media, the fluorophore retains its signal after 3 minutes. However, without the anti-fade mounting media, the fluorophore tends to fade after 3 minutes. You can see that it's beginning to turn dark. Okay, so to maximize the signal retention when you observe under the microscope, I'll give you a few tricks. So you can decrease the exposure time under the objective lens. So in this diagram, you see the objective lens. You can rotate the lens away from the sample, set up your microscope correctly, and then rotate the lens back to the sample. This will help you to reduce the amount of time imaging the sample. This may be intuitive, but it is very important to consider. The next trick is to decrease the light intensity from the microscope light source. The light source here is a white light, so you can always lower the intensity of this light. But the compromise is that the fluorophore will not be able to emit at maximum signal intensity. And just to describe the diagram here, this is a basic diagram of fluorescence microscopy. You can see there's a filter set called the excitation filter, emission filter, and dichroic mirror. The light source excites light reflected by the dichroic mirror, and then it goes to the sample on the slide. And then emission light from the fluorophore is emitted to the eye for visualization. So next, I'll go over how to maximize your signal-to-noise ratio during storage. This is when you archive the immunofluorescence slides for later imaging. First, you can store the mounted or cover-slipped specimens away from light. Just make sure you cover up the specimens in the box and store it properly. For liquid anti-fade multimedia, you may want to seal the edge of the cover slip with nail polish. And you may also want to consider this for hardening anti-fade media too. You can make sure you store the archived slides in the refrigerator at 4 degrees Celsius for extended storage. This will help to slow down the rate of photo bleaching and minimize any retraction, bubbles, and other artifacts in the sample. You can also store the slides in the freezer at -20 degrees Celsius. So let's summarize, how do I select the appropriate anti-fade multimedia? So this is a nice chart to determine which multimedia you should select. If your specimen is less than 10 microns thick, such as a cell monolayer or a thin cut tissue section, you can use a setting or curing multimedia. If the specimen is more than 10 microns thick, such as a chamber slide or well format, use a non-setting media or a liquid anti-fade mountant. And if you're considering visualization of the cell architecture, such as the nucleus, the vector shield multimedia are supplied with or without the counterstain. And this could be a DAPI. And other considerations will be multiplex immunofluorescence. If you're using various fluorophores, such as green or red, make sure you use the correct anti-fade multimedium for your application. And if you want to store long-term or archive, you can choose one of the newest multimedia for long-term storage, probably for more than a couple of weeks. And so here's a summary of our vector shield multimedia. So these vector shield media are compatible with more than 130 fluorophores. Our products have been cited in more than 60,000 references. This is more than any other anti-fade multimedia on the market. Our other product is the TrueView autofluorescence quenching kit. This is effective against non-light perfusion sources of autofluorescence. in comparison to lipofuscin, which is an aging pigment. Okay, I'll conclude my presentation, and I'll be available for any questions you may have. Thank you.
Thank you, Byron. It's really great and very interesting presentation. And we would like to discuss and explore more on immunofluorescence after this. Tapi sebelumnya saya ingatkan bagi para peserta dimohon untuk absensi dan juga mengisi feedback form yang melalui link yang sudah dikirimkan di chat masing-masing. Nah, di sini kita sudah mendapat beberapa pertanyaan. Namun sebelumnya ke sesi pertanyaan kita saksikan terlebih dahulu video PT Vantagebio Scientific Solution. Tapi stay tuned karena di akhir acara kita akan ada pengundian door prize yang menarik sekali dari Vectorlabs. Jangan kemana-mana. Okay, Byron, we've got many questions coming from your great presentation here. We're already finished. The first is from Chris Hermawan. The question is it possible to apply it for experiment using Caenorhabditis elegans and worm model?
That's a good question. So for C. elegans and other models, yes, so make sure the sample is thin cut and properly fixed, not overfixed or underfixed. And make sure you determine the optimal fixation method. Sometimes it's better to use alcohol or formalin. You can also consider the type of sample. So it could be a thin cut tissue section or frozen section.
Okay, so we're moving to the next question from Dewi Sukmawati. Currently, there are many staining kits that include the universal blocking. What do you think about this kit when compared to the blocking serum, the conventional method, which is suitable to the animal where the origin of the antibody in reducing the background?
Okay, good question. So the first part is about the universal blocker. So there are various blocking agents out there. For one is the normal serum that tends to block Fc receptors. Other agents include BSA, bovine serum albumin, and casein. They all have different effects. BSA may block charged molecules, but generally you can use different kinds of protein blocks for your application. And you may need to experiment each protein block just to see which one is optimal. What was the second question again?
Second question is, what do you think about this cake when compared to the blocking serum, the conventional method, which is suitable to the animal where the origin of the antibody in reducing the background, for reducing the background?
Okay, so like for example, if the origin of the primary antibody is mouse, make sure that the secondary antibody is not raised in mouse. Typically, it could be goat. And then from there, you match the host species of the secondary antibody with the normal serum. So in this case, because the antibody is raised in goat, you use normal goat serum. That will provide you the optimal blocking ability.
Okay, so the source of the antibody must be different, right?
Yes, so you must immunize the animal that's different from the host of the primary antibody.
Okay, great. The next question is, what is the difference between formaldehyde and paraformaldehyde for tissue fixation? Which one is the best? Does it use for different application?
Actually, they're very similar. So that's a good question. It's pretty subtle. So formalin and paraformaldehyde are different types of aldehyde fixatives. Formalin could be 10% normal NBF, normal buffer formalin. Paraformaldehyde could be typically 4% paraformaldehyde. There are some subtle differences in chemical structure, but for the general intensive purposes, you can use either one. And you just have to optimize the fixation time for each application.
Okay, Byron, I think it's enough for the Q&A session, although it's very interesting for us. Thank you so much for your explanation and troubleshooting. So, sekarang waktunya untuk door prize. Oh, sorry. We still got time here. We can still have another question. The next question from Hani Dewi. What caused the background is to hide or even the same color with the cells? So I repeat it again.
You repeat it again?
Okay, from Hani Dewi. What caused the background is to hide or even the same color with the cells? So it's difficult to document it.
So for example, if you're saying the color of the cell is green and you definitely want to avoid using a green fluorophore. Some cells express green fluorescent protein or GFP. So make sure you use a different fluorophore that doesn't overlap in the emission spectra with the green channel. So for example, for a green cell, you can use a red or even far red fluorophore to visualize your target antigen. That will help you to really provide excellent contrast between your cell and the fluorophore.
Okay, great. So the next question is, how long can the ImmunoFluorescence slide be stored for us to review it again on another time? How long can we store the fluorophore signal? How long can it stay, especially the signal?
Yeah, that's a good question. And the answer really depends on your application. There are various factors that could affect how long you can archive or store the mounted slide. So first is the type of fluorophore you selected. In general, a FITC or rhodamine fluorophore would not have as much photostability and it will photo bleach faster over time. So you may want to use a dialite or Alexafluor 488 or 594 instead, and that will really help you to increase your photostability. The type of format of your sample also matters. Could be a foam fixed tissue section or frozen section, and the archiving time does vary between these formats. After all, the frozen section can be more fragile. And so generally, the fluorophore should last at least several weeks we've done some testing, and typically it will last probably closer to 8 weeks given standard parameters and adequate storage. Of course, make sure you store the slides away from light and keep them at 4 degrees or even minus 20 degrees Celsius. The colder the temperature, generally the slower the rate of photobleaching and artifact formation.
Okay, it's really interesting. It depends on many factors, correct? So we're moving to the last question, I think. What are the advantage and the disadvantage of one-step detection and also the two-step detection methods? And based on your suggestion, which one is better?
Can you repeat the question one more time?
Okay, wait. What are the advantage and the disadvantage of one-step detection and the two-step detection? And which one is better?
So that also depends on the target antigen you select. In general, if your target antigen is strongly expressed and you know that it's going to be very abundant in your sample, then you could stick with your one-step method. The advantage is that it's a faster protocol. You just apply your primary antibody followed by your fluorophore conjugated secondary, and then basically mount and cover slip. You want to choose a two-step method if your antigen is weakly expressed or the expression is unknown. So because you want to increase sensitivity when the antigen is not abundantly expressed in your sample. So for the two-step method, there are more steps involved. You might need to use a biotin evidence system or a non-biotin system with a amplifier antibody. But the extra steps really increase the sensitivity of your signal and maximizes signal intensity for visualization. Thank you.
Next question from Ari Prasetya. Question is there any standard concentration for antibody diluent used in immunofluorescence? What factors we should consider when choosing antibody diluent?
That's a good question. So there are various ways to dilute your antibody for immunofluorescence or immunohistochemistry. For immunofluorescence, there aren't too many differences in determining what's the optimal diluent. Some people like to just use PBS or TBS buffer to dilute, but oftentimes you want to add a blocking agent such as 5% normal serum or 5% BSA, for example. So those protein blocks added to the diluent will really minimize background staining. And in addition, it will also help you penetrate the antigen. The antibody occasionally will have trouble accessing the antigen in the sample. But once you add the normal serum or the BSA, the antibody will more easily access its epitope.
Okay, so continuing the question from Chris Hermelin about C. elegans. C. elegans is very tiny and transparent. Usually, we do not need to make session of the worm. So what do you think on this case?
So typically, we have more experience with other types of tissue samples, such as thicker embryos or just thin cut tissues. So I would say our experience with C. elegans is very limited, and you may need to consult the literature for details about this organism.
Okay, then we'll move to the next question from Muhammad Yunus. Any provide monoclonal antibody for every Eimeria sp in poultry so that easily to detect infection, particularly each Eimeria sp?
Okay, so it's the question about a chicken primary antibody? You said poultry.
Yeah, poultry.
Okay, we don't have very much hands-on experience with this type of chicken or poultry antibody. We do offer a couple of secondary antibodies that are anti-chicken IgY, which also cross-react with chicken IgG. I'm not quite sure what the rest of the question means, but you're free to provide more details.
Okay, so the next question is, from Diah Prabhawati Ratnani. The question is, what is the most difficult and frequent cause of failure in preparation of an immunofluorescence examination?
So the question was about what's the most common cause of fading in immunofluorescence?
The frequent cause, the frequent.
How frequently?
The most difficult problem we're facing in immunofluorescence.
Okay, so the most difficult problem would probably be the autofluorescence background. For many years, researchers saw a lot of background from various sources, but for autofluorescence, it was very challenging to quench or mask. In the past, if there was a lot of lipofusion in brain tissue, for example, people tended to use home brews or some commercial solutions such as Sudan Black B. However, Sudan Black B would create residue on the tissue and obscure part of the signal. So in more recent years, we've developed a new quenching reagent called TrueView. The TrueView is an effective reagent for non-lipofusion sources because some tissues like kidney or spleen have high levels of autofluorescence from red blood cells or aldehyde fixation. So we've found a better solution called TrueView that really quenches this autofluorescence and makes the immunofluorescence application more applicable to investigators, and it really improves the signal to noise ratio by really dampening the background.
So we need to pay more attention on the autofluorescence, right?
Yes, that's a very common issue for this application, and we have a solution to address it.
Okay, the next question from Ani Riani. Can IF be used for detection of rabies virus in infected brain cells or tissue? Using
about a virus?
Yeah, rabies.
Sorry, can you spell that?
Rabies.
Rabies.
Yes, rabies virus.
Oh, rabies.
Yes.
We don't have very much experience with rabies virus. I would say for viral research, some viruses have certain antigens and that could be like glycosylated. And we do have detection reagents that can detect the glycosylation. We have these plant lectins that bind to specific carbohydrates in these viral glycoproteins. So that's one possible way to research. The other way is to just use a primary antibody that targets the viral protein, like the nucleocapsid or other proteins, and then from there you use your standard immunofluorescence technique, such as a fluorophore conjugated secondary antibody to bind to the antigen, which could be a viral protein. But if you want more details, I would consult specific papers about the rabies virus.
Okay, great. The next question, wait a second. From Rahma Fitri Hayati. What is the general quenching medium or reagent to block autofluorescence? The general.
What is the general
quenching medium?
Oh, the mounting medium?
Quenching.
Oh, quenching medium.
To block the autofluorescence.
So some of the common quenching reagents would be True Black or Sudan Black B. Those are only good for quenching lipofuscin. Lipofuscin is typically found in neural tissues due to aging of these tissues. But there are other sources of autofluorescence that could be from extracellular matrix proteins like collagen and from red blood cells. You can always perfuse the tissue with PBS to get rid of the red blood cells. But in case that's not possible, you can use the TrueView quenching reagent to quench red blood cell autofluorescence.
Okay. Next question from Muhammad Basharuddin. Ineffective where specific antibodies or analytical techniques? Sorry for Mr. I'm moving to the next question from Yesi Amelia. Is there update study on immunofluorescence in detect SARS-CoV? This is an interesting question.
Sorry, what type of scope?
Detect SARS-CoV, coronavirus.
Oh, SARS-CoV-2.
Yeah.
Yes, so there have been many journal papers that have used these detection reagents for SARS-CoV-2 research. Typically, they target certain primary antibodies against spike glycoproteins, nucleoproteins, and other SARS-CoV-2 viral antigens. So you can purchase these primary antibodies from other vendors and then use standard detection reagents to target and visualize these viral antigens. As discussed, you can use one-step or two-step methods to visualize the localization of these antigens. You can always do multiplex staining with these viral antigens to find the pattern or distribution in, say, kidney tissue, lung tissue in particular, and even the brain. So there's a lot of research being done on SARS-CoV-2 using just the detection reagents. And you can also use plant lectins to target viral glycoproteins too. There are certain mannose binding lectins that bind to the spike glycoprotein. And there's some studies about how they could be used for antiviral research because these lectins have antiviral effects by interfering with the ACE2 and RBD interaction, the receptor binding domain. So there's a lot of good research being done and still ongoing.
So it's still many research on SARS-CoV. Okay, the last question from Aziz Fuadi. Can this immunofluorescence be used for detection of blood parasites, especially for trypanosomiasis?
That's a good question. We have limited experience with that organism. So let me explain. So most of our internal testing is on thin cut tissue sections from humans, and mostly from common organs like kidney, spleen, and brain. So we don't have very much hands-on experience with this application, but I'm sure there are journal papers that do describe this organism.
Okay, Byron, thank you so much for the session. It's really interesting and very useful for us to troubleshoot problems we face in laboratories. Thank you for spending your Friday night with us. So, thank you.
Thank you.
Oke, baik, sekian presentasi dari Byron. Terima kasih kepada pemirsa semuanya yang sudah menyaksikan video ini. Sampai jumpa di presentasi dan video kami selanjutnya. Salam sehat dan salam sejahtera bagi kita semua. Terima kasih.